Almost every human cell contains a full set of DNA. However, not all of the genes encoded are transcribed and translated. In determining the genes to be expressed, it is important to understand the different levels of gene regulation. Though there are some similarities, prokaryotes and eukaryotes regulate genes quite differently. Because prokaryotes usually live in quite unpredictable environments, it is necessary for them to have a regulation method that helps them adjust to the environment quickly. Prokaryotes usually regulate genes through operons, which are units of length genes. There are two typical types of operons, lac operon and trip operon. Lac operon is an inducible operon. This means genes regulated by lac operons are expressed only when needed. A repressor is usually bound to the operator upstream of the lac operon, which prevents transcription factor from binding to the transcription initiation site and thus prohibits transcription. Because transcription is regulated, this is a transcriptional level regulation. It saves the most energy and resources because it shuts down the pathway at the very beginning. Take the lactose level in E. coli as an example. There are three proteins involved in the pathway: beta-galactoside permease, beta-galactosidase, and beta-galactoside transacetylase. When E. coli encounters an environment that has glucose, the three proteins are not expressed at very high levels. However, when there is a lactose in the environment, the three types of proteins start to be expressed. This period, when the three proteins are being transcribed into mRNA and then translated, is called the lag period. Within 10 minutes, the level of the proteins increases significantly and becomes ready to break down lactose. In E. coli, the lac operon is not transcribed unless a beta-galactosidase is present and predominates. At this moment, the repressor will be bound by an inducer, in this case, allolactose, which changes the conformation of the repressor and releases it from the transcription factor binding site, allowing the RNA polymerase to bind and carry out transcription. Similarly, a trip operon also can be turned on and turned off. However, the difference is that it is usually turned on. The trip operon is turned off only when needed. Trip operons also have a repressor. However, the repressors only binds the DNA when the co-repressor binds to it. Take tryptophan as an example. When tryptophan is present for a certain level, the repressor binds to the DNA, and tryptophan serves as a co-repressor in this case. In both lac and trip operons, the regulatory protein is a repressor. However, the regulatory protein can also be an activator. Activators can regulate both repressible and inducible systems. However, different from repressors, activators cause an increase in transcription instead of switching on and off the transcription. Therefore, activators and repressors can act on one gene at the same time. The combination of the effects determine the rate and whether a gene is transcribed. In addition to repressors and activators, there is also a group of proteins called the sigma factors. Sigma factors can bind to RNA polymerase and signal them to bind to specific promoters. The sigma factor is a huge advantage for prokaryotes to adjust to the environment because it is capable of signaling different pathways in a large scale, causing specific prokaryote pathways to make changes at appropriate times. Different from prokaryotes, the eukaryotic genome is usually way larger and is enclosed in the nucleus. Eukaryotes are also more complex than prokaryotes in development. Therefore, there are more ways eukaryotes regulate its gene expression. The most energy and resource-saving gene regulation level is the transcriptional level. In order for eukaryotic genes to be expressed, the RNA polymerase II needs to be bound to the DNA first. However, in order for it to bound, various general transcription factors need to be bound first. There is a region upstream of the start site of transcription in eukaryotic genes called the Tata box. It is a region that consists of mostly A and T base pairs. The protein TFIID binds to the Tata box first, changing its own shape as well as the shape of the DNA. Following TFIID, several transcription factors bind to the complex, which prepares the RNA polymerase II to bind. These transcription factors can be activators or repressors. If the transcription factors are activators, the DNA sequence is called enhancers. On the other hand, if the transcription factors are repressors, the DNA sequence are silencers. Just like the prokaryotic gene regulation, eukaryotes also use sigma factors to regulate large sets of genes that may be far apart and even on different chromosomes. Similarly, sigma factors also have great importance in responding to the environment. This is especially important when an organism is in a stressful environment, for instance, drought. In plants, there is a regulatory sequence called the dehydration response element, DRE, that will be bound by a specific transcription factor during droughts led by sigma factors. Moreover, the eukaryotic gene is usually organized into chromosomes, which are complexes that consist of histone-wrapped DNA. Therefore, it is possible to regulate gene expression through histone. Each histone has tails that are made of about 20 amino acids, and that sticks out of the chromosome complex. Usually, the histone tail is positively charged, and the DNAs are negatively charged due to the phosphate group in the DNA structure. Therefore, there will be ionic attraction that causes the histone tails and the DNA to attract to each other, making the DNA inaccessible for expression. However, an enzyme called histone acetyltransferase can bind to transcription factors and RNA polymerase, which adds acetyl groups to the positively charged amino acids, neutralizing the charges of the histone tails. This change in charge weakens the attraction between the histone tail and the DNA, which loosens up the compact, making DNA accessible for transcription. Another class of proteins called histone deacetylase can remove these acetyl groups added and thus repress transcription. Besides the acetylation of histone tails, transcription can also be regulated through methylation and phosphorylation. Histone phosphorylation takes place during chromosome condensation in mitosis and meiosis, while methylation takes place on lysine residues on histones. Depending on the different types of lysine residues that are methylated, the gene expression can be activated or repressed. DNA methyltransferase can catalyze the reaction of methylation, and DNA methylation usually takes place on a cytosine that is next to guanines. These methylation sites are usually called CpG islands. Methylated DNA will be bind to proteins that repress expression. Therefore, if a DNA is highly methylated, it is not expressed. Here comes a question: what happens when the DNA gets replicated? Due replication, maintenance methyltransferase will catalyze the formation of new 5-methylcytosine in the newly replicated DNA. Of course, similar to other gene regulation mechanisms, methylation is reversible, and the methyl groups formed can be broken by demethylase. One special feature of DNA methylation is that it is heritable. However, it is worth noticing that DNA methylation can change over time due to environmental effects. Now we are done for the transcriptional level regulation in eukaryotes. We move on to the next level, mRNA level regulation. One interesting feature of gene regulation is alternative splicing. It is a mechanism that one single gene can be cut into different mRNAs during the mRNA processing process. This process is when pre-mRNA is turned into mRNA by cutting off introns between exons. The 5' cap and the poly A tails are also added in this process. Sometimes, however, a few exons will be cut with the introns, which creates variation for the product mRNA. The difference in mRNA will thus be shown in the final protein product after translation. A few typical examples of alternative splicing would be human B cells and T cells in the immune system. Besides eukaryotes, alternative splicing is also used by viruses like HIV. HIV genome encodes 9 different proteins, but only one is translated. The gene for all 9 proteins are transcribed into pre-mRNA, and alternative splicing enables the extra genes to be cut. Moving on from mRNA regulation, the next level is the regulation of translation. There are various ways translation is regulated, which includes activity of microRNA, modification of 5' cap, and the translational repressor proteins. MicroRNAs are tiny RNA molecules. Each microRNA is about 22 base pairs long. They are first transcribed into a long precursor, Then they're folded onto themselves. The single-strand precursor becomes double-stranded. After a few more in-between steps, they turn into single-stranded microRNA molecules. The microRNA is complementary to certain parts of the mRNA and is guided by a protein when attaching. When it binds to the mRNA, it degrades the mRNA. Of course, besides microRNA that directly degrades the mRNA, there are other ways translation can be shut down. Since the 5' cap added to the 5' end of mRNA is necessary for translation to take place, modification of the 5' cap can prohibit translation. For instance, tobacco hornworm moths keeps the egg cells with unmodified GTP caps before fertilization. However, when the egg becomes fertilized, the caps become modified and thus carry out translation. Finally, in the translational level gene regulation, there are also translational repressor proteins. These proteins are capable of binding to mRNAs and thus keeping the mRNAs from binding to the ribosome. For instance, ferritin can be regulated by translational repressor proteins. When the level of iron ions increases in mammalian cells, the ferritin level also increases quickly. This is because some of the free-flowing iron ions bind to the ferritin repressor and change its conformation. This, in return, allows the translation of ferritin to proceed and enables the produced ferritin to store the toxic free-flowing ions. This concludes the translational level gene regulation in eukaryotes. The final regulation level is the protein level. Different molecules stay in the cell for different lengths of time. In order to regulate this length, proteins can be targeted and degraded. In the cell cycle, the cyclins that regulate the proceeding of the cell cycle are broken down in proteasomes. To direct protein into proteasomes, a protein called ubiquitin is needed. Ubiquitin is bind to lysine residues and proteins that need to be degraded. After one ubiquitin binds, more ubiquitin will be bind to the bound ubiquitin, forming a polyubiquitin chain on the protein. This complex will be bound to proteasomes, which are large protein complexes that have the shape of a gyro. While the protein polyubiquitin complex is put into the proteasomes, the polyubiquitin chain is removed and recycled with the consumption of ATP. Inside the proteasome, the protein becomes hydrolyzed. So this concludes all levels of gene regulation in both prokaryotes and eukaryotes. Now let's review all the different levels of gene regulation. For prokaryotes, LAC and TRP operons can switch on and off transcription. Regulatory activators can be used to increase transcription. Sigma factors are also used to direct RNA polymerase to bind to the specific promoters. For eukaryotes, the transcriptional level includes regulation of transcription factors, histone acetylation, phosphorylation, and methylation. Alternative splicing regulates the final form of mRNA. Alternation of 5' caps, microRNA, and repressor proteins regulates translation. And finally, proteasomes and ubiquitin together regulates the time a protein stays in the cell.